Hey, how's it going, sir? So this is the video for question 2, explaining the process of protein synthesis. So this is just a page of the DNA sequence that you gave us. So you have adenine and thymine and all the other nucleotide bases connected to each other via their hydrogen bonds. So the first process, or the first process of protein synthesis is you're gonna have RNA polymerase bonding to an area on the DNA sequence, usually where there's a region where there's a lot of TNAs or thymine and adenine connections because of the lower degree of hydrogen bonding, making it easier to break and unzip. So after that, after RNA polymerase binds to a part of the DNA, it's going to unzip the DNA so that the commencement of the mRNA can start. And after the mRNA strand has been fully developed, it's going to look something like this. So instead of having thymine on the mRNA, it's going to have uracil, because mRNA doesn't have thymine nucleotides, it only has uracil instead. So this is what it's going to look like after all. And once the whole strand has been developed, it's going to reach a termination point and it's going to be let go from the DNA strand. And once that happens, it's going to have a poly A tail attached to it to prevent nucleuses from destroying it or ripping it apart, and also a methyl cap to prevent any digestion of the new strand. And it's also going to play a role in the initiation of translation. Also, the exons and the introns are going to play a role. So exons are areas on the strand that are expressed, they have gene expression, and they need to be kept and linked together. And introns have no expression, so they have no use, and they're going to be taken apart by spliceosomes, and they're going to be just ripped apart, and the exons are going to join to make one solid strand of mRNA. The mRNA is then going to leave the nucleus and go into the cytoplasm, and this is what it's going to look like. So down here you have the full mRNA strand, right? Obviously it's going to start off with AUG because that's the initiation codon. So then a tRNA is going to, a ribosome unit is going to attach to the beginning of the sequence, and a tRNA is going to deliver a amino acid to eventually develop a fully formed polypeptide, which can then be folded into a functional protein. So the tRNA is going to bind to the P site, where methanone is going to initiate the development of the polypeptide, and more tRNAs are going to continue delivering amino acids until the strand until it's over. So in this case, I have the sheet right here. So there's 1, 2, 3, 4, 5, 6, 7 amino acids that need to be delivered, or actually 6, excluding the stop codon. So methanone, so this sequence is going to be comprised of methanol, histidine, alanine, isoleucine, glycine, and proline. So that's going to be the polypeptide that eventually gets folded and gets made into a fully functional protein. So yeah, that's pretty much it. Thanks.